So, we've said that all living things on planet Earth can be divided into three domains: the bacteria, which evolved first, the archaea, and the eukarya. We also said that the bacteria and the archaea combined are referred to as prokaryotic. Prokaryotic organisms have certain features associated with them. They are single-celled organisms, and they are microscopic. So prokaryotes meet our definition for microbes. The defining characteristic of prokaryotic organisms is that they don't have any organelles in them. So there is no nucleus or any other kind of organelle inside a prokaryotic organism. When we look at the DNA in a prokaryote, we find it in the form of a single circular chromosome. Now, there are some exceptions to that rule. There are some newly discovered prokaryotes that have multiple circular chromosomes, but the vast majority of prokaryotic cells have their DNA in the form of one circular chromosome. Now, that's different from you and I. We have multiple chromosomes, and our chromosomes are in a linear form. They're in a linear shape. Now, if you take the circular chromosome out of a prokaryotic cell, it's not in the form of a circle. It looks all jumbled up. And that's because in nature, DNA doesn't just exist on its own. It has to be folded up and compacted in a way that keeps it safe. In prokaryotic organisms, the DNA gets wrapped around a type of protein, which is referred to as being histone-like. When we talk about the eukaryotes, we're going to see that the DNA is wrapped around histone proteins. In prokaryotes, the histones are not really exactly true histones. Instead, they are sort of a precursor to histones, and we refer to them as being histone-like. So the DNA, again, gets wrapped around these histone-like proteins, and then the whole thing gets carefully folded up into a structure that we call a nucleoid. This is not a nucleus. This is not a membrane-bound organelle inside of prokaryotic cells. This is just a free-floating combination of DNA and protein. Now, one of the things that makes prokaryotic cells most interesting, at least in my opinion, is that there is some DNA that is located in these cells that is not found in the nucleoid. This can be thought of as extra chromosomal DNA. These occur as small circles of DNA called plasmids. And if you know about plasmids, you know that plasmids give prokaryotic organisms extra characteristics. One of the most interesting is the ability to resist antibiotic drugs. So plasmid DNA is little circular pieces of DNA that aren't part of the nucleoid. They are free floating in the cytoplasm. You don't need plasmid DNA to survive. Plasmid DNA gives cells extra characteristics that help them survive in a dangerous world. Now, when we look at the DNA in the laboratory, when we take that circular chromosome and that plasmid DNA and we look at it in the laboratory, a couple of things are very apparent. The genome is much simpler in a prokaryotic cell than it is in a eukaryotic cell. Now, what do I mean by genome? The genome is simply the collection of DNA that we find inside an organism. That's all that term means. The collection of DNA. So your genome would be very different from the genome of a bacterial cell. You would have many more base pairs in your DNA, and you would have many more genes in your DNA. Prokaryotes have many fewer base pairs and many fewer genes. For example, in an E. coli cell, and E. coli is a type of bacteria, there are about 4.6 million base pairs in that circular chromosome. Now that seems like a huge number of base pairs, but remember, in 4.6 million base pairs, there's probably about 4,300 genes. Now compare that to a human. In humans, we have about 3 billion base pairs in our genome, and we have somewhere in the order of 30,000 genes. So yes, our genome as eukaryotic humans is much more complex than the genome of a much simpler prokaryotic cell. So eukarya, the domain eukarya, is where we find eukaryotic organisms. And remember, we said the eukaryotic domain is more complex than either the bacteria or the archaea. And by complex, I mean there is a much greater diversity in the structure of the organisms that you find in the eukarya domain. In other words, most bacteria look pretty similar to each other. There are some differences, but not tremendous differences. Most archaea look pretty similar to each other. In comparison, when you get into the eukarya domain, you see tremendous variation in how organisms look. The animals, the plants, the fungi, and the protists all look very, very different from each other. Now, the protist group can be further broken down into 3 categories that I want us to be familiar with. It's in the protist group, the protist kingdom, where we find the algae, the protozoa, and what are called slime molds. Now, this is a microbiology course, and we're not going to be talking about multicellular organisms, but you should be familiar with the types of organisms that exist in the protist kingdom. Eukaryotic organisms, unlike prokaryotic organisms, can be single-celled, but they can also be multi-celled like humans are. Eukaryotes can be microscopic, but they can also be macroscopic, visible with the naked eye, in other words. So that means that some eukaryotes would fall into our definition of microbes. There are some eukaryotic organisms that are single-celled and microscopic. But there are other eukaryotes that are not microbes because they are multicellular and they are macroscopic. Just as we said the defining characteristic of prokaryotes is a lack of organelles, eukaryotes have organelles, including that important structure called a nucleus. Eukaryotes have multiple linear chromosomes, and the DNA in those chromosomes is wrapped around histone proteins to create what we refer to as chromatin. Chromatin, that's the form of DNA that you find in the nucleus of a eukaryotic cell. Now, most eukaryotic cells do not have any plasmids, but some of the most primitive single-celled eukaryotic organisms have plasmid DNA, just like their prokaryotic cousins. As we said a minute ago, eukaryotic organisms have a much larger and more complex genome. They have more base pairs and they have more genes. Remember, we said humans have 3 billion base pairs in their genome and somewhere in the area of 30,000 different genes. Let's take a minute now and think about the prokaryotes as individual domains, the bacteria and the archaea. All prokaryotes, we said, are single-celled and microscopic. When we think about the bacteria, though, we note that the plasma membrane is surrounded by another layer of material, what we refer to as the cell wall. So around the plasma membrane, there is a material, a carbohydrate called peptidoglycan. And peptidoglycan makes up this structure that we call a cell wall. Peptidoglycan is unique in nature. It is one of the strongest materials that we know about. It resists all kinds of forces that might be applied to a prokaryotic cell that might damage it or kill it. So this cell wall that surrounds bacteria is really protective and helps the bacteria survive in a dangerous world. The bacteria divide using a process called binary fission. Binary fission is a form of asexual reproduction. The parent cell, the parent bacterial cell, goes through binary fission and divides to create 2, what we call daughter cells. Those daughter cells are essentially clones of the parent cell. They have the exact same DNA as the parent cell has. Something else to know about the bacteria is that they are capable of using very diverse sources of energy. In other words, unlike you and I, who rely on organic materials to survive, and by organic, I mean carbon-containing materials. We like to eat plant products, we like to eat animal products. We need to consume organic materials to get energy. The bacteria have a much more diverse menu. Some of them, yes, some of them use organic molecules for energy, but there are some bacteria that are capable of using inorganic molecules. In other words, things that don't contain carbon. Things like, for example, rocks. Yes, there are bacteria that can consume inorganic material like rock. You and I can't do that. There are even bacteria that are capable of using sunlight as their source of energy. So just like the green plants, there are bacteria that are capable of performing photosynthesis, harnessing sunlight for food, for energy. If you compare that to the archaea, there are a lot of similarities, but there are some important differences. In archaea, there is no cell wall outside of the plasma membrane, but there is a wall-like structure. It's what we refer to as an S layer. The S layer is just structurally a little bit different. It's not made out of that peptidoglycan material, but other types of protein and carbohydrate mixed together. Bacteria use binary fission for reproduction, and so do the archaea. But there are some archaea that use other mechanisms of reproduction. For example, there are archaea that can use budding to reproduce. And all that term refers to is the production of daughter cells right off of the surface of the parent cell. You may have seen images of yeast reproducing by budding. Some archaea are capable of another form of reproduction called fragmentation. So the archaea just have a greater diversity of their reproductive processes than the bacteria do. But similar to the bacteria, the archaea use very diverse sources of energy. Just like the bacteria, they are capable of using organic materials, inorganic materials, and some species can even use sunlight. One of the most remarkable things about the archaea, and something I want you to be familiar with, is this. The reason that the archaea are so different from the bacteria is that the archaea have evolved to make use of what we sometimes refer to as extreme environments. The archaea have managed to take advantage of niches in our environment that other organisms simply cannot survive in. The archaea have evolved to be what we call extremophiles. They love extreme environments. Now, what do I mean by an extreme environment? The archaea are capable of living, for example, at pH levels that would kill other organisms. If you go around the country and around the world, there are natural locations where the pH is quite low, quite acidic. You would not find other living things living in that kind of an acidic environment, but you can find archaea living there. There are places where the concentration of salts is very high. Again, that would kill most living things, but the archaea have evolved to be able to survive in those kinds of salty environments. There are places that are very, very hot on the planet or very cold on the planet, and those extremes of temperature would tend to kill most living things. But the archaea, again, have evolved to be able to survive in those places. So, if you remember one thing about the archaea, I want you to remember that they have evolved to live in some of the most extreme environments on our planet. On this slide, there are some images that were taken underneath powerful electron microscopes. And I included this so we could take a look at this outer sort of wall structure that we find around prokaryotic organisms. If you start on the upper left, what you're looking at here is just a portion of a bacterial cell. So we're at such a high magnification here that we can't even see the entire bacterial cell. We're just looking at one little region of it. The cytoplasm is right here. The plasma membrane is surrounding the cytoplasm. They've got it labeled as PM. And then you can see there's this darker structure surrounding the plasma membrane. That's the cell wall. Bacteria are surrounded by a cell wall made out of peptidoglycan. Now, if you come over here to the images on the right, this is what an S layer looks like. Now, these images were taken with a different kind of electron microscope than these, or this image was taken. This was taken with what's called transmission electron microscopy, and these were taken with what's called scanning electron microscopy. The difference with the scanning type is that it shows us the surface details. I want you to look at the surface of this archaeal cell, this S layer. It almost doesn't look real. It almost, to my eyes, looks like this cell is covered in an ace bandage. Can you see all the little dots, all the little sort of indentations on this surface? It's very ordered, it's very structured, but it almost looks synthetic. It doesn't really look natural. It doesn't look like the natural surface that you would see on a cell, but it is. It's called an S layer, and it does provide a wall around the plasma membrane, but it is not a true cell wall because it's not made out of peptidoglycan like this one is. So in this image on the bottom, this is actually an image that they took and they manipulated to turn it into what looks more like a drawing. Again, we're not looking at a whole bacterial cell here. We're just looking at a portion of it. But I put this in because there are some bacteria that not only have their peptidoglycan cell wall, but also have a second membrane. These are what we call gram-negative bacteria. So let's orient ourselves here. We can see cytoplasm here. We can see this first structure, which is labeled as cytoplasmic membrane. That's just another word for a plasma membrane. Then you can see a very thin black line, and that's been labeled peptidoglycan. That is the cell wall around this cell. And then outside of that black line, there's a second membrane. It's labeled as the outer membrane. So there are some bacteria that have a cell wall, but also a second membrane. Those are what we call gram-negative bacteria. Now I put this slide in just to drive home the idea that the Archaea like to live in extreme environments. What you're looking at in this slide are some actual images of these extreme environments. Up at the top here on the left, this is a location in Yellowstone National Park that's referred to as the Morning Glory Pool. This is not colorized, folks. This is what this pool of water actually looks like. The blue color that you see in the middle comes from all of the archaeal organisms that live in the water that fills this crater. The organisms themselves give the water that bluish color. And I think you can just make out that there's steam coming off of this pool, and that's because this water is very, very hot. This is the kind of temperature that would kill most living things. Bacteria would not be able to survive in this kind of heat, but the Archaea can. Over here, we also see a very hot environment. You can see steam here. But in this image, we're actually on the bottom of the ocean. We're on the floor of the ocean, and we're looking at these structures that vent the terrific heat that comes from the center of the earth. These are called thermal vents, and they're found at some of the deepest parts of the ocean on the floor. The Archaea love these kinds of extreme environments and survive well in this extreme heat. Finally, I included this image. This is a picture of a river in Spain. It's called Rio Tinto. It's extremely, extremely acidic. It's also full of heavy metals, which is why it has this very unusual sort of orangey rust color. Not a lot of things can survive in that combination of acid and heavy metals. But there are archaea that live here and do very well there.